Good afternoon, everybody. We just had a really nice lunch. I hope you had a chance to talk to some friends, maybe reestablish some connections that have been missing for the last year since the last one. Whatever. Anyway, what we're here to listen to is Biology Without Borders. And were any of you aware that there's a video? Eileen isn't with us today because Eileen broke her leg. Hello, Eileen.
Eileen is with us today. Eileen is there. There's Eileen.
She's here virtually. But she sent me what they call their elevator video. And it's a very nice overview, and I'm sure you'll share the link with them. So we'd like to hear all about it. The toolkit, I always love tools. There are times where you say, I wish I could do this, but I don't have the right tool. So Pamela Borowski and Karen Kackley they're all three, and Eileen are from Lehigh Valley, and they've made this trip here to share it with us.
Thank you. Yes, just before the presentation, somebody said to us, break a leg. I said, no, we don't do that. We got one that broke her leg already. We're not adding any more to that one. So what I'd like to talk about today is basically an instructional toolbox that I use in teaching biology courses at Penn State Lehigh Valley. And it's basically to kind of think outside of the box. And basically that box is the classroom. And I want to get them outside of the classroom and have instruction take place both indoors and outdoors. But there's many different tools that I'm using to do this. So I'm using everything from open source materials to some strategies that are basically, quite frankly, non-tech. All kinds of apps that I've found for, I love my iPads, and digital textbooks, video, software, hardware. So we're going to cover kind of a gamut of the different types of tools that I use in my biology courses at Penn State Lehigh Valley. The main course that I feel the greatest responsibility for is Biology 110, Freshman Biology. And this is a core course that is absolutely critical to the biology major. And so there's supposed to be somewhat of a consistency throughout the different campuses on this course. And one of the things that I'm finding is that I have a particular set of challenges with this freshman biology course. The first thing is that biology, unlike many of the other sciences at Penn State, has no prerequisite for Biology 110. So there is no math prereq, there is no English prereq, there's no chemistry prereq. So oftentimes we may have students coming in who are not very well prepared, and they can't take their chemistries and they can't take the maths that they need, but they can take that biology course. So there they are. So I don't have any way of screening who comes into the course. And it's a quite complex technical subject that requires a lot of understanding of big concepts. And I have diverse biology experience coming in here, meaning that in one semester I can have someone who may be a provisional student through someone who had a 5 on the AP Bio exam and has credit for Bio 110, but just wants to make sure they're not missing anything. So I've got that full gamut that I need to serve. And as one of my 5 on the AP exam students said to me, you know, you can't ignore us good guys either by putting too much focus on those that their skills need to come up to do well in there. And then there's a whole diverse learning style issue. So many students are visual learners, others are tactile learners. So it's a way of trying to deal with many of these different learning styles that the students have. And here's a perfect example. We depend a lot on microscopes in Bio 110. I don't see anything. Okay, you put that sample under that scope, and some students can find it right away. Other ones, they're not saying anything. They're just sitting there and they're sitting there and they're sitting there, and you see no response, and you come over and you find that like the microscope slide isn't even under the lens. It's like completely off to the side, but they can't see anything at all on it. So it's this whole range of skills, and I try to tell them it's like riding a bicycle. You know, the more you do it, the better you'll get, and eventually you'll get to that point. But it's often a challenge with these students, and they feel terrible, they feel stupid, because they don't know how to use the equipment yet. So I find that it really puts up a barrier for them. So there's a couple of different things that we were able to do this year that were quite successful, with some interesting hardware and some lab videos and some apps that we found for use on the iPads minis. So one of the things that I'm attempting to do in this class is it's called Basic Concepts and Biodiversity. And if I take a lot of my freshman students and I have them look at this campus and I say, what do you see? They're going to tell me a building and grass. Well, there's a lot more going on biologically on that campus than a building and grass, but they may not see that. And so what I wanted to do was to get a way to get them outdoors, particularly in the fall, in touch with nature, in touch with biology. And the project that we had used previously was a herbarium project. And I had the students go out and collect different plant materials, and this was a very long and laborious project that was very messy because they had to get leaves. I asked them not to collect on campus because I didn't want our trees denuded, but they had to go out and get different plants. They needed to press them. There was a matter of having enough presses for 60 students. With all of these things, they didn't know how to identify them. The idea was that I was going to teach them how to use a dichotomous key and how to learn how to identify different plant materials and learn different plant features, but then there was the issues of gluing, and it was a very long and hard project, and then grading all of these things was particularly difficult. And then came out this wonderful app called LeafSnap. And then I found out that all of my ideas kind of went out the windows because the students learned that they could take an iPad, photograph the leaf, and identify it. So my whole idea of making them learn the different characteristics and work through a key had just kind of gone out the window. So we decided to try another way. And here at the University Park campus, they were utilizing this lab called Bioblitz. Now this is actually a nationwide program. It's kind of one of these citizen science things where people go out and they look around them and they look for insects, they look for plants, and they look for many of these different things. But one of the issues that I was running into was the equipment that was required. Okay, so Vanna, if you would, Vanna White is going to demonstrate for me the equipment that students in Bio 110 at University Park were taking into the field with them. They had to check out a laptop computer and, let's see, look at her skills, that's wonderful. And one of these microscopes with a light source, and then they could go out and they could take photographs. But this meant carrying around a laptop, computer, and a microscope and checking them out in order to do that. So even though I wanted to do this laboratory and take them out on campus, I thought this is not really going to work very well for our purposes. Until one day, my magical lab technician was shopping around online and found a microscope that fits onto any iOS device. And this is one that's ready to be hooked up to a, the microscope itself fits on any iOS device at all. This is one that's set for an iPhone 4S. This is one for the full-size iPad, and then this one that you have in front of you is for iPad minis. And it comes this full set of equipment for $149. Now you can buy additional, what do I want to say? The docking device is $20 to get interchangeable. So you can get more than one of those. So we got a couple of these to just look at and play with, and we were so thrilled with them that we wanted to get more. So we went to the chancellor and we said, well, here's the problem that we have. We don't know what devices the students are going to come in with. So we got a little grant and we got 10 of the iPad minis, which are here. So that way I knew what device I could dock them onto. The beauty of this is it just uses the camera on the phone. And so you're going to get to play with these. But we can take photographs right through here, right on the camera. There's no focusing required. And what we did was we had each student go out into the field and they needed to find 2 examples of flowering plants, non-flowering plants. For those of you that aren't botanists or biologists, that'd be like mosses and ferns, things like that. 2 vertebrate animals, 2 invertebrate animals, 2 fungi, and then 2 things that we're just going to call microscopic organisms. And they had to take photographs of these, and then they would bring them back in and they did a PowerPoint presentation about their organisms. So they had to find out all kinds of information about what family they were in, where did they live, what other organisms did they interact with, just to give us a little kind of oops, okay, reconnect to Doceri. Oops.
It's a picture of the moth.
We lost the internet connection, so I'm going to need to go back to the mouse. But anyway, they they had to find these different organisms, and we got some excellent photographs. And the other thing that I found was the immediate engagement that these students had. We had no learning curve at all. So there was none of this, like if I brought in pond water and had them look at different organisms under the microscope, I have the whole issue of can I use the microscope or can't I use the microscope? Immediately, you put it on and you see. The squealing out in the field was totally amazing. They were just thrilled with what they were seeing. And the other thing that I found that was really interesting was that having that iPad, having that physical barrier between them and whatever it was they were looking at, took away fear. So people that normally would be afraid of spiders were like right on top of a spider or right on top of ants. And they were just out in the field and they were digging and they were having this wonderful time. So they felt immediately engaged in the material, and they just said this was one of the highlights of their semester, was using these. And so they could come and check them out at any time that they needed to go out and finish their collection. And then they worked on these projects and did a presentation on their different topics in the Bioblitz. And the other wonderful thing about this is we have apps on there that are free that take the place of field guides. So when they're going out and they're trying to identify it in the field, these were all free apps. Eileen can laugh at me. I don't pay for any apps yet. I'm so proud of myself. I've had all these years of having an iPad and I've yet to pay for an app. So we have the Leaf Snap app, which you take a photograph of the leaf and it identifies the plant for you. We have a free Peterson bird guide, which has the common birds of Pennsylvania. Nature Tap, which has other insects and other trees. And then we have other things we've used on there like iCell and molecular modeling. But all of these things are available on the iPad at no charge. So they can go out, they have what they need to identify it, they can go out and they can take the photographs and they can be outside enjoying nature and team building, all the things that just came with this that were unexpected bonuses, like that being engaged, having the team building with the other students in the class, and then not feeling stupid because they can't use a microscope. And now that microscope does come later. I mean, they do have to learn that skill, but at least this helps get them started. Then we started realizing there were a bunch of other things that we could do with these mobile microscopes. And they do a lab where they take Daphnia, which are little water fleas, and they're looking to see the effect that things like sugar, alcohol, caffeine have on the heart rate of this little water flea. And we found that these scopes worked out quite well. And then the other thing is that we found a free app that replaced what was the cost of a lab counter? Do you remember? It was over $100, right?
I think so.
And so they could sit there and they can tap, like every time they're observing a heartbeat, they could just tap on this counter to see how fast the heart rate is. So rather than counting 1, 2, 3, 4, 5, 6, 7, they just have to tap, 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 tap. And then they have all of the data. So we're still finding the things that we can use these iPad minis for. We haven't even scratched the surface of the possibilities of what these can be used for. Another thing that we've done with them is to record lab procedure introductions. So particularly when we're doing labs like this one that we're showing here, where they were doing bacterial isolations and looking to see about whether the ingredient triclosan, which is in antibacterial soaps, to show that it basically, we've all got resistance to it now, and it's not doing the beans good on us, but it's certainly hurting the ecosystem. But they were doing bacterial isolations. If I'm standing in front of the room and trying to show you how to do something on a Petri dish this large, it's pretty difficult. But what I can do here is I can record it ahead of time. I can have it up there. First of all, they should watch it before they even come to class. And then the other thing is we can set these iPad minis up around the room at the different stations, and if they've forgotten what to do, they can hit the play button again. So we've been using them to film these lab procedures. Another thing that we used the iPad minis for, just as a matter of fact, last week, a week ago, and today I was still in Mexico, but we sent iPad minis with students that went to Mexico, and they took photographs and they blogged every day about their experiences in Mexico. And we put these up on a student blog, and so they would, photograph what they saw, and they had questions that they needed to answer regarding, like each one had a different subject that they were studying while they were down there. And they had to post every day at least 2 photographs and 2 paragraphs about what they'd learned on their subject during that trip. Okay. Open source materials. I feel sorry for students because of the costs associated with many of these materials that they need to use. So I found a lot of open source things that I use, as well as going to digital textbooks. Textbooks cost entirely too much. Okay. Here we have Pearson's Campbell Biology, the standard that's used in most courses. I don't know what it's up to now. I think it might be $300, closing in on $300, I think, maybe, for this, with Mastering Biology. The students don't read it. It's way too heavy. I kid them, if you want to take it to class, you get a flat tire on the way home. You can stuff it under the axle, change your tire. And so I just didn't feel that they were getting the benefit out of these textbooks. So I wanted to look for alternative methods for getting them the material. So about $300 for that. I have gone to a book that comes, if you're familiar with the journal Nature, which is a very standard journal. They now have a book called Principles of Biology by Nature Publishing. It's $49. The students have lifetime access, including all of the updates. It links to journal articles in the primary literature, so I can take them out to primary literature articles. They can look at that. There are little quizzes that they take, reading quizzes as they go through, and it really is born to be an online book. It's not a textbook like Campbell's Biology, which they took and they made it into an ebook. I mean, it really is. It's written in little modules. It's written to be used digitally. And you can get it, you can download it on your iPad, you can have it with you, they can bring it to class. And the interesting thing about it is when you open up the book, the figures come alive. So if you want to see the process of photosynthesis, you can click on a chloroplast and watch the process happening. So it really is born as a digital product, not simply to be an ebook. Now that's what I use for the majors course. And I don't mind having them spend $49 for that book. For the non-majors biology courses that I teach, I use completely free textbook. OpenText, this is funded by, it's run out of Rice University, but they've got a lot of big donors, like the William and Flora Hewlett Foundation, Bill and Melinda Gates, and that sort of thing. And these books are available online for free. They can download them onto as a PDF. They can put them on their computer. They can do anything they want. And then you have that student that says, I don't like digital textbooks. I want a paper textbook. Here it is. $29. 5 days later, it was in his mailbox. Printed version of the textbook. And you can look through here, and if any of you are biologists, you'll see it looks just like any traditional, typical text, biology textbook. Has all the main material available with wonderful illustrations. And as I say, it was like under $30, and he had it in 5 days in his mailbox. Because at our campus, the cost of textbooks can be prohibitive for some of our students, and they're not getting their books until way late in the semester. And biology is not the only subject that they have. They have physics, sociology. The regular biology is for majors, but I still like my nature book because of the other features that it comes with. But A&P, chemistry, stats, calc, econ, psych, and they're just expanding, because they were finding that access to classes wasn't so much a hindrance anymore as it was to the actual materials that were needed to teach the classes. So I've gone away completely from the doorstops to the digital, and it's no longer an issue. If they want a printed book, they can get it. I use a lot of free quality online resources. EdPuzzle is a wonderful thing. You can take any video that you want, put it on EdPuzzle. You can insert questions at any point that you want. You can stop, you can make commentaries, so you can go onto TED Talks or whatever and get a video, and you can stop it and put your comments in at any particular time. You can put in questions. The students go in and log in, and you see all of their responses. They're there for you. I use things like, we've probably all heard of the Khan Academy videos. And then there's a great one called Crash Course Biology, which I like. The guy's really funny. And then this group education portal, they're basically like a MOOC. And so you have these courses, you go on and you take them, and then, but all of their materials are free. And so you can use these wonderful little modules that they have. And believe me, I have used all of these this semester because we had, I don't know how many snow days this semester. I think in one class I missed almost like 9 hours of class time because of delays and snow. So sometimes I would record a little lecture in Screencast-O-Matic, a 15-minute thing, put in questions as we go along to test their concept knowledge. Or use these other little things, and I have the quizzes, and I can watch whether they did them or not. So it's mainly for completion that I'm doing those. Okay, then some non-tech strategies that we're using, and some software. Okay, I was lucky enough. The Schreier Institute for Teaching sent me to a Lilly conference this past year, and Eileen and I both went. And boy, if you get the chance to go to Lilly, jump on it. It's amazing. And I saw an author there, Kathleen Gabriel, and she talked about teaching unprepared students. And so I've gone through and I've looked at a lot of the information, read this book. In fact, we had a learning community on our campus based on this book. She's such a compelling person. She herself was a provisional student who now is a provost, I think, at Chico State or something like that. So she went from being someone who, as a freshman, had an English teacher say to her, "What are you doing in this classroom?" to someone who's now a provost. And her whole thing is on teaching unprepared students. And so many of them come in and they're just really not ready. So there's many things that we can do. We can use clickers for formative assessment, things like concept maps for scaffolding knowledge. Should I say the word schema here so that you'll smile at me, Eileen? She loves schemas. She even taught me what they were. I do things like I record lectures in Doceri. Now, Doceri's cut out on me for some reason, but I record all my lectures in Doceri, and I put them on Angel at the end of the day, because I had several students last semester. One was undergoing chemotherapy, and she couldn't be there. I had another student who had to have back surgery that missed like 3 or 4 weeks, and they could catch up with everything. And then I have students that just want to go back and listen to the lecture again. I hand out things like a performance prognosis indicator. I do things like drop one of the exam grades. So let's look at some of these. Okay. So the clickers for formative assessment. I usually use 2 or 3 clicker questions in there. Kind of just review, what have we gone over? Are you understanding that material? And I find they're very helpful. And again, I get very positive feedback from the students regarding the use of clickers. Concept mapping. I went to a lecture this morning that was great on a guy that's actually writing his book as concept mapping. But this is that whole idea of, there's a wonderful little thing called Lucid Chart that the students can use. It's no cost with full features. They can use it on the iPad, and they can draw these different concepts. So what we're looking at here is an example of photosynthesis. And this is like the what we call the light reactions and then the Calvin cycle. And I can look at this and I can tell that student gets this concept. And then I look at other concept maps and it has like, we can see here where these processes are related and how they come in and what's going in and what are the products that are coming out. This is an actual student map, one of the students created. And then you have other ones where it's just like a process over here and one here, and they don't see any relationship, and they don't know what's coming in, and they don't know what's going out. And then I have them work collaboratively on them. So bring in their different maps and then work together to see if they can improve them for a group map. I mentioned Doceri already, so I record the lectures and make those available to the students. So that's just an example of one of the things. And Eileen had to pick the one where the woman paid $53,000 to have her dog Booger cloned. But we were going over the fact that cloning in animals can happen. In South Korea, they'll clone your dogs for you. But the DEA is actually cloning some of their excellent drug dogs, because some have better noses than others. But anyway, I will record the lectures in Doceri, so those are available. And as we say, the prognosis performance indicator and the dropping an exam are just no-tech strategies. So I hand them this little quiz at the beginning of the semester, and I say, I'm not collecting it back, so you'll answer this truly. You answer this, not what you think I want to see. You answer this, truly for yourself. I'm going to attend all the lectures and labs. I'm going to bring my iClicker to every class. And this goes on and on. Actively engage. I'll make flashcards. I'll do all my homework. I'll teach the concepts to my dog. Any of these things. And then what I do is at the end, they flip it over. And I say, okay, based on how many of these you answered true, this is the grade you can expect in the course. And then when they come to me after the first exam and maybe their performance wasn't what they wanted, I'll say, let's bring out that prognosis predictor again. Because they're like, what do I do to get a higher grade? And I think they're thinking I'm going to give them extra credit or something. I say, let's go over this list. Let's see what you did do and what you didn't do. What can we add to your strategies to improve your performance? So, again, came right out of Kathleen Gabriel's book. Want to play? We've got iPad minis. We've got, Pam has brought all kinds of wonderful things for us to look at. And by the way, Eileen just put together this morning for us a Weebly that has information on all of these different tips. So Biology Without Borders, Weebly.com. So if you have any questions, feel free to ask them. But the main thing we want you to do is to actually play with this equipment, to use these microscopes and see what they can do. You just simply turn on the iPad and then turn on the camera function. And then on the side of the microscope, this little button here that's next to the triangle, you hold it in for a couple of seconds, and that turns on the light. So you have to press it for a couple of seconds, and then the light should come on. There you go. And then you just set it right down on whatever it is that you want to see. And you use the pinch and what do you call it? Expand? Pinch and expand? Like from your fingers for the camera function. And you immediately have an example. Okay, and if you want to use leaf snap, we have a leaf here. So that you can use like the leaf thing. I think the butterfly wings are some of the coolest things to look at. Oh, and then you can take a photo with it too. Oh, I'm not. I don't need her. Yeah, you've got a bird feather here.
Oh, you
So hold that in, there you go. And then just, is it on the camera? You're on. Turn that on. Why is it right on?
You have to push this actually like in. Push it in and then you.
There it is. That one's not turned on. And then once it comes up, then you just put it right over the thing, and then you can look at it and you can bring it. Now the thing is, you do have to put it down basically right on top of it. Basically, just almost set it on top. Well, they're broken up in segments.
What's this one?
That is a mothman.
Okay, so we got the capture.
Just the camera. Turn on the camera. No app, just turn on the camera on the device.
You knew it.
And then you're gonna see right through, it's gonna go right up.
It's smart.
So you may have to set it right down on it.
I can see it.
Yeah. And then it's detail. And if you wanna magnify, you simply take your fingers and do, what do you call it? Expand or whatever. Pinch and snap or whatever. Yeah, there you go. Look at the difference there. And so if they have insects, they can see insects. They can do.
a microscope, or do you just use the camera?
This is just a skill, right? If you don't want to use it, then you take the, out of it.
Okay.
Yeah.
So you just use the camera?
The camera?
Or the microscope?
Yeah, it just uses the camera.
Oh, okay.
That's all it does, is that there's no app required, it just uses the camera that's on the.
Boy, 20 years ago when I was a biology major, I loved it.
Oh, I know, this is what I need, and then they start feeling better about themselves, and they start getting engaged, and then they just start using all of these materials, and they just come right out of their shell.
I'm taking a picture of myself.
Yeah. Yeah.
It's not sponge.
And they're all used to using this, because they all have different smart devices. And these ProScopes can work with other devices as well. They can work with like an Android or any of these other things as well.
How hard is it to you just snap it on and do something to get it to recognize?
It just works right through the camera. You just turn the
You
camera on.
You just snap it on the bottom? And that's it? Wow.
Nothing
I thought,
required.
yeah, you would have had to install
Nope,
some
no
software.
apps, no software, no
Wow.
nothing. You just pop it on,
That's awesome.
and it takes them, and then they can take their photos with it.
This one's really pretty.
Yeah.
And then what we've done is we've set up a thing that they come back in and they download the photos onto a place where they can get them an angel. And then they can work on their presentations where they have to find out the information. Now we should do leaf snap.
Yeah, it's gonna do that.
Okay.
These here, the old version, you did have to have software to download.
Sure.
Yeah.
I want this for my cereal.
For $149,
Go out all the time.
$149.
Very reasonable.
Lumen, ProScope Mobile. Yep.
Do you know if they make them for like Kindle Fire?
I think they may have several. I don't think it's only iOS. I think they might be making them for Android and stuff.
Hold on, Eileen has an insight.
What is it, Eileen?
Hold on.
Samsung
Go ahead.
Galaxy.
Okay, the Samsung Galaxy, I think it's something S, but it's on the website, and I put the link on the Weebly.
Yes, if we go to our Weebly.
Okay, thank you.
I don't know if this will
My
come up.
my phone.
Which is that? Wanna see your wing?
I see Science Fair
So if I go here.
Is this the one here?
Yep, but just click on Leaf Snap. And then when it comes up, we need to do the photograph part. So we go home. Okay, snap it is where you take the picture.
Even so, they.
Okay, you can use the location. Okay? Let me see if I can get this to come up. Is this one?
The other one was evil.
Those things are police.
Yeah, that ruined my whole project.
So Karen, you said you have a cost of 60?
60.
And that's
That's a really good identification.
Yeah, but they meet, and here's our little Weebly that we put together. So that actually Eileen put together. So here's where all the tools are. Hey, I give credit where it's due, Eileen. Okay, so here's where all of the different the links are. You can see some of the lab video introductions, the Bioblitz lab itself. The clicker response systems, the lucid chart mapping, any of the open source stuff, and the international study she's got up here. That was the spring break trip we just took, and then the tips on the underprepared students. So that Weebly has the different things. So Pam is doing a leaf snap now. I understand they're improving leaf snap. In that right now, in order for this to work, you have to remove a leaf and put it on a white piece of paper. But yesterday I heard they're getting it so that you can actually do it on the plant. They're trying to do it on the plant. But it's very easy. She just took a photograph, and up comes the different possibilities of oak, of what it is, and it's right there, and they can do the identification of the plant right from the
I'm just loving this.
Yeah. And so they can do that out in the field. If they have a piece of white paper with them, they can go out, they can find it in the field, they can take a photograph, they can identify it.
And can you just hold the piece of paper behind the leaf?
They might be able to. It does need to be kind of flat, because what it does is it goes on the outline shape.
Yeah, hold it.
Yeah.
But that's team building, right?
Yeah, they did team building, and let's see, who has birds on here, don't we?
I think so.
Broad finder, here's Peterson, there's Peterson's bird guide.
This ISO thing is neat too, how you see it like three-dimensionally.
So you can look up like hummingbirds, all this stuff comes right up on the, on here, all free. Now, obviously, we can pay more to get more information, but oftentimes for them in the field, this is good enough. And I believe this is also one that has, this one.
And what do they do? That's what we want.
Okay, so, let's see, do we have,
What do you call this?
here's a ringneck pheasant. Where's my volume? Here we go.
So did you have to get a grant to support biotechnology, or was that out of like normal university resources?
They had the chancellor identified a donor who was looking to donate a little bit of money to the campus. Now these, see, I think, what did they cost us? Like $450 each to get a set
I
up,
think it was a
right?
$5,000 grant.
Yeah.
Wasn't that, or was it 10,000? I think it was
No,
that much.
no, I think it was $5,000. I think it was a $5,000 grant to
I'm
get
just
all
thinking
of
about
this.
scaling this up to
Mhm.
get that. in.
But see, I'm in lab sections, so I have one of these for every 2 students, because my
Yeah.
lab only holds 20. So it's not that bad that it's only 2. Because at first I was thinking if I could get 1 for each lab table, that would be great. But instead I was able to get 1 for each 2. Oh, here's a turkey.
Would
So you can-
oils support something like this? Oils?
Yes, actually, we were in the process of writing the quail grant, remember?
I'm sorry.
That's a turkey. We're gonna get the turkey going. But anyway, this even has sound that goes with it for the field. So they can sometimes identify birds by call. Because that's what I teach them in ecology as well, is that you don't always necessarily have to see an organism to know that it's there. Sometimes you can hear it, which is why birds are great, because you can tell kind of like where they are, what species they are by call. And then yes, sometimes we use like feces, to know that they've been there. Owl pellets. What are those structures on the top of it? What, the ocelli? No, those aren't ocelli. I don't know. I don't know where to go for that.
What's the question?
Oh, I don't know.
Sorry.
I don't know. We can Google it though. Now that we're inside and we have internet, we can Google it.
There we go.
So.
Very nice.
Well, they do have ocelli, which are like a light sensor, and that might be what they are on that one, I'm not sure.
And since you were talking about grants, I'm from the Schreier Institute for Teaching Excellence, and our grant call is open
There you go.
right now. So if you have a project that you want to propose
Wow.
before April 15th, send a note to site@psu.edu, and then you get set up with one of us, and we
Get right,
talk
Eileen.
with you.
You're home, get a head start on these guys.
There you go.
But, you know, it's just amazing what it is when you put these things in their hands, what, you know, you find you can do. We thought we had just one use for them, and then it's just turned into multiple uses. Multiple uses. So, you know, we have fun. We have fun
Maybe
playing
you get
with
them,
them.
maybe you get them excited enough that they can join a citizen scientist.
Well, that's one of the things that we're gonna try to do this semester is we're working on a phenology project, which is the study of when things break bud or when birds show up and that sort of thing. And it's actually going into a database that's helping to track climate change. So in certain areas, birds are coming back sooner, plants are leafing out sooner, and that sort of stuff. So they're going through this process of, we're gonna take them out, have them learn to identify the trees. So we're gonna go back to that, okay? But they have to learn to do it when they're like that, not when they have leaves on them. So we'll be going over, you know, twig structure and buds and how plants change and learn birds and that sort of stuff, so that they can now start adding this to the citizen science database for when these things actually, you know, when they came out, when they broke bud, they have to learn all the different stages that they need to enter to do that. So we're gonna have them do that one as well. And then I learned out some other cool stuff today too they can do. And one of the other classes where they can go in and do some transcription of scientific things and stuff like that. So, but all of these things work out great. And then they can even have their textbooks of them on these things too. My dream would be if enough people would adopt these, you know, electronic books that each student could come in and buy an iPad mini at the beginning of the semester. If enough faculty would commit to
Then it
using
would
a
be
digital
worth it.
book, it would pay for. I mean,
Yeah, it would be
I can
worth it
almost
for
that
them.
textbook is almost the cost of this right here, and they can get multiple books on this one device. But I guess that's kind of what Apple is trying to do. There's the war out there, who's going to get what. But with like open source, the OpenStax books, you can get it on any of the devices. That's Lawrence saying hello. Hi, Lawrence. You know, they can download it on any device that they want. So I'd like to see the publishers not just go towards either Apple or, you know, PC, but.
But even if they have to pay something for the textbook to get it on there, it's way cheaper than Kindle forms.
Yeah.
Florence. You said all
Hi,
of
Florence.
these are, all of these actually on your website?
I think most of them are. I think she put most of them on the weekly site. So, but I just go onto iTunes and or to the App Store, and I just type in biology, and then you can do it by price, and I just look free, and then I just go through and I find what I did. So we haven't paid for any of these. Now, if I wanted a better bird book, I'd have to buy right to Peterson. But I mean, this has cardinals and robins and the basic. I mean, you'd just be surprised. I guess maybe you wouldn't be surprised, but I was surprised at the nature deficiency that our students have. You know, when I say to them, "Oh, go outside," and you find like, you know, moss growing in a moist area. Huh? Moss, ferns. I mean, they don't know what
They don't
deciduous
even know what
means.
it
They
is.
don't know any of this stuff. So just getting them out with these things and having them, you know, collect is a lot of fun. The only downside was I had to make sure they didn't get in the poison ivy. Because they did get very excited and they'd start tromping through things, looking through mushrooms and stuff, and I'm like, ah, yeah, get out of there. So we learned PI right off.